How does COVID-19 mRNA vaccine work? By Jonathan and Gabi. In 2020, vaccines for COVID blew out previous records, going from development to approval in just a matter of months. They were driven by billions of dollars and global effort.
Vaccine against coronavirus.
And it's incredible how quickly this vaccine and others have been developed.
This was done in record time.
This is extraordinary. This is the fastest
vaccine.
Vaccines are used to prevent diseases can otherwise cause death. Think of them as instructors that teach our immune system how to respond and fight back against bacteria, viruses, or any other foreign microorganism that may enter our body and cause a disease. These organisms are also known as pathogens. Currently, there are mRNA, DNA, and protein vaccines in the market, but today we'll just focus on mRNA vaccines.
Though it might seem surprising for some of you hearing this, the truth is that mRNA vaccines, just like those from Moderna and Pfizer BioNTech, have been in development for over 20 years. Compared to other vaccines that use whole viruses, either attenuated or killed, the mRNA COVID vaccine only uses a small part of the virus, occurring on its surface with the form of a spike, hence the name spike protein. This protein is the one used by the mRNA vaccines to produce antibodies and cells like B cells and T cells to produce the immune response. At this point, you'll probably have a lot of questions. Let me explain. COVID-19 vaccines simply produce the same immune response that happens in a natural infection, but without actually infecting your body. It sounds easy, right? Let's go a little deeper.
Isolating and preparing spike proteins for a vaccine will be a lengthy and costly process. Researchers had to come up with an idea. What if our own cells could actually produce them? We just have to give them the blueprint and they'll do the rest. Scientists found a way to get the mRNA that codes for that protein, but there's a problem. mRNA is very delicate and can degrade easily. How do we take it into the cells? Well, we'll be using a lipid nanoparticle, a phospholipid bilayer that can encase mRNA and serve as a vehicle for it to enter the host cells.
Take into consideration that the mRNA will code for the spike protein mentioned at the beginning of this video, derived from SARS-CoV-2 virus. The nanoparticle will be delivered into our body through an intramuscular injection and enter our cells. But don't fear, this mRNA will not be involved in any way with our DNA. It will use our ribosomes to make proteins in a process called translation. After this translation process, the proteins will be expressed on the cell membrane into two types of proteins, MHC2 and MHC1. MHC1 is found on all nucleated cells, whereas MHC2 is only found on immune cells like lymphocytes, macrophages, and dendritic cells.
The T helper cell, also known as Th, has a particular membrane protein that interacts with the shape of the S protein presented by MHC2 molecules, named TCR, and also has another protein that interacts with the MHC2 complex called CD4, activating the Th cell. At this point, the Th cell will release substances called cytokines, such as interleukin-2, interleukin-5, and interleukin-4, to tell the B cells to multiply and differentiate into these type of cells called plasma cells. We're almost done. Don't worry. Plasma cells will make antibodies targeting the S proteins to the actual SARS-CoV-2 virus. If the antibodies attach into the protein on the virus, the infection will be prevented, causing no further damage to our body. Cytokines will tell our T cells to multiply and create memory cells that will help in future infections.
Don't forget that vaccination, face masks, social distancing, and hand washing are key to fight COVID-19 virus.